During the first years of life, the mammalian brain operates like a highly plastic sponge. It rapidly forms trillions of new synapses, absorbing massive amounts of foundational data about the world. Yet this intense period of data collection is eventually wiped completely clean. As adults, we experience a near-total void of episodic memory prior to age 3.5. We retain language, habits, and motor skills, but the autobiographical record, the specific events and experiences of our earliest years, is entirely missing. For much of the 20th century, the dominant explanation for this blank space was purely psychological. Sigmund Freud argued that infantile amnesia was the result of repression, an active defensive mechanism designed to hide heavily charged psychosexual experiences from the conscious mind. That theory breaks down when we look outside of humans. Infant mice, rats, and other mammals also exhibit infantile amnesia, despite lacking the complex psychological conflicts that psychoanalytic theory requires. If a mouse rapidly forgets its infancy just as a human does, infantile amnesia is a universal mammalian hardware limitation. It requires a physiological explanation rooted in the architecture of the brain, rather than a psychological defense mechanism. People often push back against this biological limitation, claiming to hold vivid, accurate memories of events from their first few months of life. The cognitive science consensus points to a different reality. As detailed in the scientific literature on childhood amnesia, these highly specific earliest memories are actually reconstructive fictions. One case study from this research features an adult claiming to remember an event at exactly 2 months old. They described sitting outside their parents' dark green Morris Minor, noting the car was on the left side of the road, 4 meters away. A 2-month-old infant brain lacks the spatial, semantic, and categorical frameworks to encode concepts of a meter, distinguish left from right, or identify a specific car model. The adult brain has retroactively built a narrative out of later knowledge and fragments of stories told by family members. Since conscious, explicit memory of early infancy does not exist, the biological mystery remains. We need to determine whether the infant brain simply fails to encode the data at the time, or if it successfully encodes it and then physically destroys it. The initial assumption is often that the infant biological hardware is at fault, that the hippocampus, the brain's memory center, is simply too immature to physically encode an episodic memory. To test this, researchers at Yale pioneered a method to conduct functional magnetic resonance imaging on awake infants between 4 and 25 months old, allowing them to measure neural encoding in real time. This diagram illustrates their setup. They expose the infants to novel images while measuring the blood oxygen level dependent, or BOLD, signal directly in the hippocampus. The data revealed a direct correlation. When the BOLD signal showed strong activation in the hippocampus during an infant's first viewing of an image, that infant spent significantly more time looking at the same image during a later behavioral test, proving recognition. Furthermore, the strongest encoding activity occurred in the posterior region of the hippocampus. This is the exact same anatomical area responsible for successful episodic memory formation in adults. The infant hippocampus is fully capable of encoding memories. The data is written to the drive. Therefore, infantile amnesia is not a failure of input at the hardware level. It is a retention and retrieval failure that occurs later in development. If the infant hippocampus successfully records episodic memories, some physical process must be wiping them out. That process starts with neurogenesis. During the mammalian infancy, the brain generates and integrates a massive volume of new neurons at a prodigious rate. A specific memory is stored as an engram, a physical circuit of connected synapses. When millions of new neurons integrate into the hippocampus, they form their own connections, meaning existing circuits must be forcefully broken and rewired. Studies led by the National Institute of Mental Health and the Hospital for Sick Children demonstrated this in mice. As new cells integrate into a specific region called the dentate gyrus, the physical architecture of the existing memory networks is disrupted and overwritten by the sheer volume of new growth. To prove this mechanism, researchers artificially decrease the rate of neurogenesis in infant mice. Without that rapid influx of new neurons, the mice retained their early memories, dramatically reducing the effect of infantile amnesia. The rapid brain growth required for maturation literally dismantles the neural networks holding early experiences. The amnesia is an unavoidable biological cost of upgrading the hardware. Neurogenesis explains a massive physical disruption, but does it eradicate every trace, or do some fragments survive? To answer this, neuroscientists look directly at the engram, the specific biophysical cellular trace where a distinct memory resides. A study from Trinity College Dublin revealed that infant memory engrams actually endure into adulthood. However, as this cellular diagram shows, they are actively silenced by microglia, star-shaped immune cells in the central nervous system. These resident immune cells function as memory managers. They navigate the neural network, tagging the early engrams and actively suppressing their expression so the information cannot be retrieved. When researchers pharmacologically inhibited these microglia in infant mice, the suppressed engram cells reactivated. The mice successfully recalled fearful experiences from their infancy that they otherwise would have completely forgotten. The brain's immune system actively quarantines early memories, establishing infantile amnesia as a highly regulated biological imperative. This presents an evolutionary puzzle. Mammalian biology invests massive metabolic energy to encode these episodic memories, only to use neurogenesis and immune cells to systematically bury them. The answer lies in a concept borrowed from machine learning: overfitting. As seen on the left side of this diagram, an overfitted system learns its initial training data so rigidly that it hits every piece of noise, failing to generalize to new data. A properly trained system, on the right, extracts a smooth, predictive curve. Overfitting is exceptionally dangerous for altricial mammals, species like mice and humans that are born completely helpless and dependent on caregivers. Infancy is a highly specific, narrow state of existence. If the brain permanently encoded the exact parameters of total helplessness as its foundational baseline, the adult organism would lack the behavioral flexibility required to survive independently in a complex environment. Around age 3, as language matures and the concept of the cognitive self emerges, the brain actively clears out this outdated altricial training data. It drops the noise to make room for a mature, continuous autobiographical timeline. This adaptive erasure formats the neural landscape, clearing the hyper-specific noise of infancy to ensure the development of a flexible, independent adult.